Okay, we don't want to erase all of it. Okay, so back to DNA replication. And if you haven't put that as a heading yet, you'll probably want to do that. DNA replication. So, the first thing that has to happen in DNA replication is we have to open up the DNA molecule kind of like a zipper. And maybe you'll recall that I said these are hydrogen bonds that hold these bases together, and that those hydrogen bonds are very weak. So it's pretty easy to break open those bonds and to open up the DNA molecule. So that's the first thing that happens. There's a molecule that's called helicase. Again, I'll write that word for you down. Okay, helicase. It's an enzyme that can open up the bases, break those hydrogen bonds apart. So I'm going to show that real quick. Maybe. There we go. So we're just going to open it that far. It really does unzip a lot of it at one time, but we'll just do a little section so we can keep track of what's going on. Alright, so back to our molecule. I'm going to move these down now. Okay, so your next step that you're diagramming, you want to show this DNA opened up. C, T, T, G, C, A. So the helicase has come through, it's opened up the bonds between the bases, and again remember there's sugars and phosphates on there as well. Then there's a second enzyme that comes in called DNA polymerase. Notice all these enzymes have A's at the end of them. We talked about enzymes like that, which is going to match up the bases in the correct order. Now, the way the DNA polymerase works is always from the 3' to 5' direction. So on my top strand of DNA up here, I'm going to start at the T and work my way over to the left. So with the T, I'm going to need an A. G, I'm going to need a C. C, G, A, T, G, C. Now remember, those bases are coming from out here in the nucleus to get paired up with the old DNA. On the other side, I'm going to be starting over here because this is my 3' end. 3' to 5' is in this direction. So I'm going to add G, A, C, G, T. As DNA replication goes on, it's going to open up more of the DNA molecule and continuing to copy those bases in the same order. So we're going to move on from this diagram and look at some diagrams from your book, which is a little more intricate in its drawings than this one that I've set up for you. So we'll do a little review here from these diagrams as well. So in this first diagram, what you see happening here is your DNA molecule has been unzipped. The kind of pinkish, orangish things you see here represent the polymerase. So these are polymerase. You can see that the top polymerase molecule is moving to the left, and the bottom polymerase molecule is moving to the right. Then we have names for these strands where the polymerase works in opposite directions. The strand where it moves in the same direction as the way the DNA is unzipping. So for example, this top DNA molecule is unzipping to the left. The DNA polymerase that's moving to the left, so on the top strand, is called the leading strand. And the one on the bottom that's moving to the right is called the lagging strand. Now the reason they're called the leading and the lagging strand is because that bottom strand actually does copy a little bit more slowly. It lags behind. The reason for this being is that every time the DNA opens up a little bit further, it has to backtrack in order to make its copy. So let's take a look at what I mean. Again, the DNA is opening up this way. As it opens up this way, this polymerase on the leading strand can just continue to copy in this direction. On the lagging strand, however, it copied this first section in this direction. It ended up here. Once the DNA opened up a little bit more, the polymerase had to back up to here, reattach, and then copy again in this section. So what's happening is every time the strand opens up, the DNA molecule opens up, the polymerase on the lagging strand has to detach and back up, and then again copy in the 3' to 5' direction, creating these little chunks of DNA. Here's one. Here's the second. Okay, so these chunks of DNA we see on the lagging strand are called Okazaki, again, last name of a person, fragments. Again, these Okazaki fragments are only on the lagging strand because the DNA polymerase has to detach and back up every time the helix opens further. If you look at the bottom picture on this diagram, you'll see a new enzyme come into play here. This enzyme, right here in teal, is called ligase. And ligase is the glue that sticks together those Okazaki fragments. What it's really doing is creating a covalent bond between the phosphates and the sugar. So phosphate plus sugar, covalent bond. Okay, that's what the ligase is doing. So on the lagging strand only, remember only on the slow strand, we're going to have these Okazaki fragments bonded together with this enzyme called ligase. Now we're going to look at a little bit closer what happens on this lagging strand. There is another enzyme involved, which is helpful in telling the polymerase where to start, and that's this pinkish enzyme up here at the top. And this pinkish enzyme lays down a layer of actually RNA that tells the polymerase where it's going to start copying. And so every time that polymerase detaches and goes backwards to start over again, it's always got that RNA as kind of a marker of where to stop and start. So there are little chunks of RNA on that lagging strand as well. Okay. And then another little detail to point out is when the DNA is replicated, it's not like the whole helix is opening up at once. Okay, so remember DNA is a double helix. Not like the whole double helix unzips at once. Just a little section of the helix is going to open up, and they call that a replication bubble. And then again, both sides are going to replicate at the same time in that replication bubble. So we get still a leading strand and a lagging strand. So here's our leading strand polymerase. Here's our lagging strand polymerase. Here's the helicase opening up the DNA molecule. But it's not going to unzip the whole thing at once, because behind that bubble, so back here, it closes back up after replication takes place. Alright, so the whole purpose of DNA replication was to make some new DNA, okay, to make copies of DNA to go in all of your cells. And if you look back at this picture real quickly, remember when I had the bases drawn on here, we were actually creating 2 new DNA molecules. So I'll just throw some on here again quick. ATGCC would have been paired with TACGG, and then I'll do these in a different color. Do TACGG and CCGTA. Okay, and this represents one DNA molecule, and this represents another. And each DNA molecule has 2 strands. One strand, the one in black on each one, is from the original DNA, and the other strand, the one I'm highlighting in green, is new DNA. So this type of replication is called semi-conservative replication, because half of the old DNA is saved in each new DNA molecule. So we've created 2 new DNA molecules where before we just had 1. And that new DNA, one of those 2 new DNA molecules can go into your new cell, so that cell has instructions for what it needs to do. Okay, I think I'm caught up with all the replication things. Now we're going to move on to another job of DNA, which, as I said, is to be the instructions for the cell.